Well, it's my great pleasure to introduce you James McCoy, my first graduate student, actually, and founding member of the Tonkin Lab. So this is really a special occasion for me as well. James came to the lab from the University of Melbourne, from a Bachelor of Science, Bachelor of Arts degree, and that was really obvious when I saw his first piece of writing. It was very verbose and colloquial. So we really had to beat that out of that over time. And in fact, it was when he first joined the lab. I'd already signed up one honours student. This was the first year of my faculty position, and I had one honours student, and I had already signed up one, and there was I had no money. And so I had to go to Alan begging, please let me have this honours student. I think he's really good. And by golly, that was really the right decision, I think, because he was the one who ended up staying on for the PhD. The other one didn't, and which was a bit annoying. But it did take a bit of convincing to get James to carry on. And I think that we both came to the agreement that it was the right decision for him in the end. And so today he'll present you some work that's both published and unpublished, some exciting new stuff he's been doing. And I hope you enjoy it.
Thanks, Chrissy. Now, you always remember your first, right? And we'll see how well Chris beat the verboseness out of me. So today I'm going to be discussing the role of a group of calcium-regulated kinases in the lytic lifecycle of Toxoplasma Gondii. And the image you're looking at here is actually the parasite toxo made from a movie, a Z projection of a series of images from a movie showing the parasite gliding over a two-dimensional surface. So toxo belongs to the phylum Apicomplexa, which is a eukaryotic phylum which contains a number of medically and agriculturally relevant parasites. These include Eimeria, which causes disease in poultry mainly, the Cryptosporidium species, which cause gastrointestinal disease like diarrhea, and of course the Plasmodium species, causative agents of malaria, still responsible for maybe a million deaths per year. Let's get that away. But I'm interested in the far more sexy parasite Toxoplasma Gondii. You can see it replicating away here nicely. These are obligate intracellular parasites, just like any other apicomplexan. So toxos gained a reputation as the crazy cat lady parasite, mainly due to the fact that its sexual life cycle occurs in cats, whereas the sexual life cycle for malaria occurs in mosquitoes. Here, the cat is the sexual host. And in this, humans are actually accidental secondary hosts. They're dead-end hosts for the parasite. So any infection is normally just accidental from the environment. But toxo is almost ubiquitous throughout the world. Between 40 to 80% of individuals in any one population can be infected, and it can cause severe disease in vulnerable individuals, including birth defects and stillbirth if infection occurs during pregnancy. Toxo is still the main cause of neurological disease in AIDS patients. And a number of studies have shown a consistent link between infection by toxo and mental illnesses like schizophrenia. But aside from all this, toxo is most valuable to us because it's a really good model organism to understand the other apicomplexan parasites like the malaria species. And this is partly because of the really nice conservation amongst the apicomplexans. So you can see here Toxoplasma tachyzoite and Plasmodium merozoite. These can be thought of as effectively analogous. These are both asexual replicating parasites. And you can see conservation of some of the main features which are essential to host cell invasion and continuation of the lytic life cycle in these parasites. So this includes the apical complex, which importantly for my presentation today, includes the microneum organelles and the rhopteres, which are both secreted during host cell invasion, as well as egress from host cells. You've also got the inner membrane complex here, between which and the plasma membrane is housed the actomyosin motor, which powers parasite motility. You can also see that Toxo is a much bigger parasite, which makes it just very valuable for microscopy, just in a sheer resolution sense. Toxo is also a lot easier to grow in the lab. It grows extremely rapidly and is extremely hardy. Plasmodium starts to die within 20 minutes of release from host cells. So if your Plasmodium culture has destroyed all its host cells, then it's pretty much cactus. Toxo can survive pretty much at room temperature in the bin for a week. So it's a good idea to leave it a little bit longer before throwing out the rubbish. Their life cycles, the life cycles of the apicomplexans really can be thought of as roughly analogous. And the main differences are in the host cell receptors, which the parasites recognise. So here you can see a diagram of Plasmodium parasites infecting a red blood cell and Toxoplasma infecting a nucleated cell. And Toxoplasma is really interesting in that it infects basically any cell in your body. If it has a nucleus, it can infect it. So if to look at this lytic life cycle in a little bit more detail in these parasites, and I'm sorry for that resolution there, it's really nice up here. Looking at the Toxoplasma parasite at this, and what's known is that this lytic life cycle, proceeding from replication, egress through host cell invasion, is regulated mainly by calcium signalling events. So here, while the parasites are replicating inside the host cell, they're exposed to a relatively high potassium concentration in the host cell cytoplasm, and this is thought to dampen down all kind of motile events. The parasites are pretty much just sitting there waiting for some kind of signal. And these extracellular stimuli still aren't properly understood, although I'll be discussing a little bit of that today as to some current ideas. So after some kind of extracellular stimulus is received, the parasites upregulate calcium signalling, releasing calcium from intracellular stores. And this leads to secretion of the microneme organelles, those apical organelles. This includes secretion of proteins like a perforin-like protein, and this perforin will permeabilize the parasitophorus vacuole, as well as a number of adhesins. And the secretion of these adhesins allows the parasite to engage its actomyosin motor, allowing egress from the host cell. After egress, parasites have switched from a high potassium concentration inside the host cell to the relatively low potassium concentration outside of the host cell. And once in this low potassium concentration, calcium signalling is thought to no longer be inhibited and now can stay switched on. So parasites will glide around, moving until they find a new host cell which is suitable for host cell invasion. After invasion, they're again gone from low to high potassium again, which switches calcium back off, turning off microneme secretion and turning off motility. But when we talk about this kind of calcium release and through to these, the endpoints of motor activation and microneme secretion, what's happening in the middle really isn't understood. We really don't understand what enzymes, what proteins are involved in translating a release of calcium stores to motility of the parasite, invasion of a host cell, egress from a host cell. And that's what my project, has mainly been focused on. But into this mixture has come a lot of interest in a group of kinases called the calcium-dependent protein kinases. So these are a group of calcium-regulated kinases, as the name suggests, which are found exclusively in plants and some protists, including the apicomplexan parasites. And because of the complete lack of these kinases in any mammalian genome, they're really interesting drug targets. And the CDPKs all have this canonical structure featuring an N-terminal kinase domain, which is fused to a C-terminal regulatory domain. This regulatory domain includes an autoinhibitory domain, which blocks the substrate binding site of the kinase. Here, when the kinases are exposed to calcium, calcium binds to two calmodulin-like domains, which bind the calcium, causing a conformational change, removing this autoinhibition and activating the kinase. So theoretically, as soon as you release calcium from an intracellular store, you should be able to activate CDPKs. So these are really interesting hubs of calcium signalling in the apicomplexans. And you can see that this structure solved here, and you can see the really dramatic conformational change. So this CDPK here is another Toxoplasma CDPK. This is actually CDPK1, which was shown a few years ago in 2010, actually, to be involved in secretion of the microneme organelles. And this was the first really nice piece of evidence showing the CDPKs were probably pretty important in regulating host cell invasion, host cell egress. So when I started all the way back in honours, I was interested in the kinase CDPK3. And this was chiefly because CDPK3 is the closest homologue of the Plasmodium kinase CDPK1. CDPK1 had been shown to localize here to the plasma membrane, as you can see CDPK3 does, which was thought to co-localize it really nicely with that actomyosin motor, which drives parasite motility. And a couple of in vitro phosphorylation studies had shown that it might phosphorylate this actomyosin motor. And you can see here the CDPK3 is co-localized with one of those members of the actomyosin motor, GAP45. So nice piece of evidence to start off with in honours. So we were able to make a knockout of CDPK3 pretty easily, which was kind of disappointing at first because, well, it suggested it wasn't all that important, perhaps. Obviously, it's not essential. These parasites, the knockout parasites, grow really nicely. But we performed an egress assay. And in these egress assays, what you're doing is essentially the parasites are replicating really nicely here inside the host cell, and then you add a drug called a calcium ionophore, which causes artificial release of these intracellular calcium stores, which stimulates calcium signaling, leading to egress from the host cell. And when you do that with wild-type parasites, this is measuring in minutes and seconds. So after about a minute and a half of the ionophore being added, the parasites activate motility and escape from the host cell. What we saw when we tried this with knockout parasites was that after we added the drug, the parasites essentially didn't respond at all. They stayed essentially immobile, trapped within that parasitophorous vacuole, up to and beyond the 10-minute mark of adding the drug, after which the experiment stopped. So obviously this is an interesting phenotype. And so we wanted to quantify this defect in egress from host cells. So here, just to run you through it quickly, we're just doing an egress assay where we're taking parasites which are growing inside host cells and then stimulating them with that calcium ionophore, then fixing them after 3 minutes with paraformaldehyde and performing an IFA. And you can see here, wild-type parasites, this is an egressed vacuole, this is an egressed vacuole, this is an egressed vacuole over here. Those parasites have all spread out from a central vacuole. You can see immediately there's a big difference between the wild type and the knockout parasites, whereas the knockout parasites are essentially all still trapped inside their parasitophorous vacuoles. And when I quantified this, you can see a really obvious difference in their ability to respond to this drug and escape from host cells. So the next question I wanted to ask was, how important is that plasma membrane localization to its activity? A lot of kinases are known to be pretty promiscuous unless they're targeted specifically to an area of the cell. So CDPK3 has this really obvious dual acylation motif in its N-terminus. Here, what we'd expect to see in this consensus motif, which is found in a number of eukaryotic proteins, it's not special to toxo. You have co-translational myristylation of a glycine residue at amino acid position 2, followed by reversible palmitylation at a nearby cysteine residue. And it's known from toxo and other eukaryotes that when you disrupt or when you mutate either of those glycine or cysteine residues, then you expect membrane localization to be disrupted. So I tested this in CDPK3, and you can see that this worked really nicely. CDPK3, when you mutated either the glycine residue at amino acid position 2, the cysteine at amino acid position 3, or both residues, you had a really obvious switch from a plasma membrane localization to a cytoplasmic localization. So then the obvious question is, how does this cash out in terms of activity of the kinase? So here, I attempted to complement the knockout parasite strain with these N-terminal acylation, mutation kinases. So when we complement the knockout strain with the wild-type copy of CDPK3, you see that normal egress is essentially restored. However, when you complement with the glycine mutant or the double mutant, you have essentially no rescue at all of the parasites. What's interesting is that if you mutate only the cysteine residue, although the localization is cytoplasmic, you do get essentially a complete recovery of egress capabilities. This is probably because myristylation of the glycine should still occur. Palmitylation of the cysteine is dependent on myristylation of the glycine, but not vice versa. So what's probably happening here is that a very small amount of CDPK3 is being targeted to the plasma membrane, and that's just enough to restore normal activity. So this is interesting because it's showing that these kinases are very sensitive to their localization, but also they're very active. You only need a small amount to complement this activity. So I'm trying to hypothesize a place for CDPK3 in this nascent calcium signaling cascade. We knew that we know from previous studies that, as I mentioned, in egress, you need secretion of the micronemes, which includes secretion of that perforin-like protein to permeabilize the parasitophorus vacuole, and you need engagement of the parasite motor. So we'd seen previously, well, as you saw in those movies, the parasite motor was completely unable to engage. Those parasites stayed completely immotile within the host cells. But there's still the possibility that the parasites may still be able to release their micronemes. So we need to be able to pick apart those two processes, the activation of the motor and secretion of those microneme organelles, including those adhesins and the perforin-like protein. So this is an example of a parasite which can be thought of as having defective microneme secretion. This is a parasite which has had the perforin-like protein, which is secreted from the micronemes, knocked out. And what you see in these parasites, when you perform the same egress assay, that the parasites aren't able to lyse the parasitophorus vacuole, but they do activate motility. So they stay trapped within the parasitophorus vacuole and just spin like they're in a tumble dryer, just going round and round and round. On the other hand, if you still have normal microneme secretion, so you're still secreting the perforin normally, but you don't have motility, you get something more like this myosin knockout, where you can see, I'm not sure if you can see, but I can see, that the parasites are completely trapped. The parasitophorus vacuole has basically disintegrated around the edges there, and the parasites aren't able to get anywhere. So we couldn't see any motility. Does that mean that they're also not able to release their micronemes? Oop, still going. So we transfected parasites with a copy of a fluorescent protein called DS-RED, which was targeted to that parasitophorus vacuole. And these parasites were then had motility prevented by adding an actin filament disruptor. So these parasites can't go anywhere, but they can still release their micronemes normally. So when you do this experiment with wild-type parasites, what you see is that after you add the ionophore drug, that the parasites release their micronemes, release that perforin, and that fluorescent protein disperses out of the parasitophorus vacuole. Okay. When we tried this with knockout parasites, we never saw this release of the fluorescent protein. You can see that the fluorescent protein is completely trapped within the parasitophorus vacuole membrane. So this is a really nice piece of evidence showing that CDPK3 was acting on both motility, the parasites don't move when you stimulate egress, and they also can't release their micronemes. So CDPK3 is probably acting fairly high up in the calcium signaling cascade. We wanted to confirm this looking at the display of other microneme proteins on parasites when you stimulate them. So in this assay, I've got wild-type parasites, and this is just by IFA, obviously. Here, when you don't have the drug, you can see that they're at the apical tips of parasites, you don't see any display of micronemal proteins. However, after you add the drug and then fix the parasites, you see they start to display the microneme proteins at this apical tip of the parasites. And so confirming our results from release of the perforin-like protein, again, we saw that CDPK3 knockout parasites weren't displaying these microneedle proteins at their apical tip. So while parasites are intracellular, they can't egress normally, they can't activate motility, and they can't secrete their micronemes normally. What was interesting, though, is when we performed another kind of microneme secretion assay while parasites were extracellular, so they're not inside host cells trying to get out, they're outside trying to get in, microneme secretion was suddenly normal. So you see here for wild-type parasites, what you want to concentrate here is the supernatant fraction, which is showing the proteolytic release of microneedle proteins into the supernatant of this experiment after you stimulate them at various points, by various means. So here you're stimulating microneme protein release. And comparing wild type to CDPK3 knockout, we saw that release of these microneme proteins was now suddenly exactly the same between wild type and knockout parasites, in complete distinction to what we'd seen for parasites which are trying to egress. But what about motility? Do extracellular parasites also activate motility normally? Well, yeah, they seem to. This is an invasion assay, which I won't go into the details of here. But here, knockout invasion is being normalized to wild-type invasion. And as opposed to that egress assay, where you saw that really clear difference in the egress capacity of parasites, here you can see that invasion is essentially one-to-one. And if there is a difference, it's not particularly significant. So again, it seemed to be suggesting that while parasites are inside trying to get out, they knockout causes a real problem. But if they're outside trying to get in, CDPK3 has no role whatsoever. Finally, looking at a gliding assay. So this is actually that image that I showed you earlier of parasites gliding over a 2D surface. So this is what you'd expect to see after egress and before invasion. While parasites are swimming around trying to find a new host cell suitable for host cell invasion. And so after you've done that movie, you can take all your images from the movie and make that Z projection and assay how many parasites are motile. You know, that one's obviously a motile parasite there, and that's an immotile parasite there. And again, when we did this, there was no difference between wild type and knockout motility, either without stimulation or with stimulation with that calcium ionophore drug. So all that data was showing that CDPK3, as I mentioned, is only important while parasites are trying to egress from a host cell and is completely dispensable when they're outside of host cells already. So we wanted to ask whether it's the local potassium concentration which is important for CDPK3's activity. So I mentioned earlier that when toxo is replicating within the host cell, it's exposed to a relatively high potassium concentration, which is thought to inhibit calcium signaling. Now, you can actually stimulate egress of toxo just by partially permeabilizing the host cell membrane and the parasitophorus vacuole membrane using a detergent called saponin. So here, when you add the saponin, the potassium which is inside the host cell floods out, dropping the potassium concentration. Parasites suddenly have their calcium signaling switched on, and they can egress normally. And you can see this here, where wild-type parasites, when you expose them to low potassium concentration, either without or with the additional help of that calcium ionophore drug, that they egress as you'd expect. Whereas knockout parasites, suddenly they have a partial restoration of egress capability. It's not back up to 100%, but it's also not that complete dropout of egress, which I showed you in that first figure. So is this due to the potassium concentration changing, or is it due to the loss of membrane stability when you add saponin? So here you can, if you expose parasites to a high potassium concentration outside, in the extracellular media, you're not having any drop in the potassium concentration, egress stays inhibited. And you can see that here, where compared to the previous figure, if you don't give the parasites the extra oomph with that calcium ionophore drug, the wild-type parasites are essentially inhibited in egress if you saponin treat their host cells in a high potassium concentration. And here, much like our previous figures, you can see that the CDPK3 knockout parasites are still completely inhibited in their egress. And adding the calcium ionophore drug does very little for them. So what this seemed to be suggesting was that, yeah, there was something to do with the local potassium concentration, but it was a little bit more complex than that. We weren't just getting a yes or no answer with the egress capability of these parasites. And so what we suggested in the paper that this was published in at the end of 2012 was that actually CDPK3, the loss of CDPK3 led to some kind of reduction in efficiency of egress. And that's what CDPK3 is actually doing is acting to amplify existing calcium signals, which trigger egress from host cells, gliding motility, and reinvasion of host cells. In this, CDPK3 might be important in allowing parasites to rapidly escape from a host cell. So if parasites can't really quickly get calcium signaling going and escape from a host cell, they might be more prone to attack from the immune system, something along those lines. So in this model, CDPK3 would only be important here in the switching from a high potassium concentration to a low potassium environment. Once parasites are already extracellular, CDPK3 no longer has any function. And here, the idea would be that CDPK3 is important for a rapid upregulation in the level of calcium signaling after some kind of egress stimuli is received, such as a drop in potassium concentration. So CDPK3 would be acting really nicely here to really ramp up calcium signaling. And without CDPK3, you still get these kind of calcium signals slowly ticking over, but you don't get that really nice rapid upregulation. So and that was the model we were left with. The CDPK3 is phosphorylating some protein, probably in the plasma membrane. Remember that its localization is essential to its activity, which is acting in some way alongside other possibly redundant calcium signaling cascades to activate the actomyosin motor, to activate microneedle secretion to allow parasites to egress from host cells. So at the end of that paper, we were interested in alternative pathways of egress. You know, if there is some redundancy in these systems, where does it lie? And how much redundancy is there? So it already known that another kinase, protein kinase G, which is a cyclic GMP-dependent protein kinase, is also essential for egress. And this has been found through small molecule inhibitor studies. But you can also activate egress of parasites by stimulating them with a cyclic GMP phosphodiesterase inhibitor called zapranast. So what phosphodiesterases do is they break down cyclic GMP. If you add zapranast, the breakdown of cyclic GMP is inhibited, and you get cyclic GMP levels building up and up and up, allowing activation of calcium signaling. Oops, spoiler. Okay. So, how much redundancy is there in this system? It had been suggested that this kind of PKG signaling can overcome the need for CDPK3 if you're activating PKG and not CDPK3 using the zapranast rather than that calcium ionophore. And we saw the story was a little bit more complicated than that. When you add the drug zapranast, yeah, again, wild-type parasites do rapidly upregulate egress, and knockout parasites don't rapidly upregulate egress, and that's, in case you can't make out, that's 2.5 minutes, not 25 minutes. But after a certain period of time, the knockout parasites actually catch up. You know, they get there in the end. So again, this was suggesting that CDPK3 is acting in that amplification loop to rapidly upregulate some kind of initial calcium response. And PKG is just acting to activate that initial calcium response, and then its own pathway gets going. Oh, and I just described that there. Yeah, so PKG and CDPK3 do act along the same pathway, but again, CDPK3 is probably acting as some kind of amplifier. At the same time, we were interested in another kinase called CDPK4. CDPK4 also localized the plasma membrane, and we're again able to knock it out very easily, but as far as we could tell from all previous assays we've done, it didn't do anything. It didn't have any role in egress or invasion or anything. However, when we now tried these zapranast experiments on it, we saw that these CDPK4 knockout parasites had a kind of a halfway defect. At this early time point, the CDPK4 knockout parasites are kind of half as efficient as the wild-type parasites, although they do also eventually catch up. So the obvious question is, what happens if you make a CDPK3/4 double knockout parasite? And if you do that, then you see parasites are completely kaput. They can't respond to the Zaprinast stimulus at all. So what this suggested to us is that there's actually a really complex overlap in the calcium-dependent and the PKG-dependent signalling pathways, and there's probably a role for multiple CDPKs at multiple stages of these pathways. Here, we might think that PKG stimulus of calcium release does have to go through CDPK3 and CDPK4 and maybe another kinase and maybe another one on top of that. And CDPK4 and CDPK3 here are really acting synergistically to rapidly upregulate activation of the actomyosin motor, secretion of the micronemes to allow the parasite to activate egress. But to change tacks slightly, we still, at the end of that, still didn't really know much about what these kinases are acting on. And that's really what we're more interested in, is actually what is the nature of these pathways. The kinases are all very well and good, but you can see that we can knock them out pretty easily and do these assays pretty easily. So we could just kind of we could just knock them all out, and in fact we've tried that, just knocking them all out and doing these assays. But what's the actual nature of the pathways themselves? We wanted to try and establish the substrates and interacting partners of these kinases. And one way we thought to try and do that is through random chemical mutagenesis in toxo. So here, what we thought to do was to actually attempt to recover rescue of egress mutants from a CDPK3 knockout population. So here, what we wanted to try and do was take our egress defective parasites and randomly mutate them to recover parasites which had rescued a normal egress phenotype. So here we take our knockout phenotype and treat them with the chemical mutagen EMS. And I'm only showing one population here at a time, but actually, when I did this experiment, I did it in 10 populations simultaneously. You then take your parasites from your populations and stimulate them with the calcium ionophore. When you do that, you'll activate, most parasites will still have the parental CDPK3 knockout egress defective phenotype. However, some mutants might have recovered rescue of egress phenotype. So you can recover those parasites, wash them away, and you can repeat this over and over to enrich for these parasites. And after about 2 months of trying this on 10 different populations, we saw that for two of those 10 populations, comparing wild-type parasites to knockout parasites to these mutant parasites, we actually had 2 different populations which both produced clonal parasites which showed some rescue of egress. And these are the populations, population 7 and population 10. Now these are just, 2 clones from each population. So this is really exciting, obviously, that we'd been able to perform this really speculative mutagenesis study. So we isolated 3 clones from each population and submitted them for whole genome sequencing at the AGRF. When we got those sequences back, what was really cool was that actually from the clones within each population, they all had an identical phenotype, suggesting not only had only 2 of our 10 populations rescued, but within those populations, the rescue population had arisen from a single mutated parasite. So this is a very rare mutation. And between those 2 populations, one hypothetical protein was mutated independently in both of them. And that was this protein, a hypothetical protein at the time, although only slightly less hypothetical now, which we've given the creative name of TM5 because it has 5 transmembrane domains and nothing much else to speak of. So looking at the localization of TM5, you can see that it localizes, again, like CDPK3, like CDPK4. It also localizes really nicely to the parasite plasma membrane. When we look at the mutations that had occurred within these populations, we could see that within population 10, we'd had some parasites had introduced a stop codon right near the N terminus, suggesting that within these populations, mutation of TM5 was effectively destroying its activity. So we tried to just do a simple knockout of TM5 in wild type and CDPK3 knockout backgrounds. And when we did this, we saw that knockout of TM5 in wild type parasites essentially had no effect on egress. But when we knocked out TM5 in the CDPK3 knockout, the double knockout, we saw exactly like the clones which we'd recovered from the mutated populations, again, saw the lovely rescue of egress. So this was really exciting. We really hit the nail on the head, actually. So work is still continuing with TM5 to understand what it's actually doing, what its role is in Toxoplasma. But what we think its essential role is as some kind of suppressor of egress signalling. Here we imagine that under normal circumstances, when that extracellular stimulus is received, which leads to release of calcium from an intracellular store, activating CDPK3, this CDPK3 might phosphorylate TM5, removing some kind of inhibition that it holds on egress, allowing the parasites to escape from the host cell. However, in the absence of CDPK3, where you can't get phosphorylation of TM5, parasites remain inhibited in egress. So, and there is some evidence to date to show that TM5 might actually be a direct substrate of CDPK3. And this has been done using a process called the Substrate Captor technique. So here, this is just to run you through it very quickly. This is a schematic of CDPK3. And when we look at the ATP binding pocket of the kinase, it has what's called a bulky gatekeeper residue, in this case a methionine. And this gatekeeper residue prevents bulky ATP analogues like this inhibitor here from accessing that ATP binding site. However, when you mutate that large methionine to a much smaller glycine residue, you can suddenly make these parasites sensitive to these kinds of inhibitors. But what you can also do is make them sensitive to ATP analogues such as thiophosphate, which allows thiophosphorylation rather than the normal phosphorylation of a kinase's substrates. So here the mutated analog-sensitive kinase can thiophosphorylate its substrates, and then after the addition of an alkylating agent which targets that thiophosphate site, you can immunoprecipitate your thiophosphorylated substrates. And in a bit of preliminary and unpublished data to date, one of our collaborators has shown with quite nice confidence 3 potential phosphorylation sites on TM5. So in work going forwards, we're going to try and use phosphomutants of TM5 and to try and complement that double knockout capability. If you put in a phosphomutant version of TM5 into the double knockout background, is it able to, again, stop parasites from egressing, or do the parasites still egress because TM5 is still non-functional, essentially? But aside from genomics, we'd also like to go deeper into proteomics to try and characterise these calcium signalling pathways in Toxoplasma gondii egress. And for this, last year, I was lucky enough to be able to go to Vancouver, Canada, to the University of British Columbia, and work with a group there who make really nice use of a process called SILAC with quantitative phosphoproteomics. And here the really nice genetic flexibility of Toxo really shone out, allowing us to make use of our knockout parasites in these phosphoproteomic studies. So here, just to give you a quick rundown of the strategy, you'll have 3 cultures growing independently. You might have your wild-type population, your CDPK, well, just to run you through it again, wild-type population, which has normal egress, CDPK3, TM5 double knockout population, which has restoration of egress, and CDPK3 knockout population, which is still defective in egress. And these are all grown in the presence of either light, medium, or heavy stable isotope-labelled lysine and arginine. We can then stimulate egress in each of these populations separately, produce lysates and mix the samples, and we then enrich for phosphopeptides using first strong cation exchange for fractionation, and then enrichment using tiox for phosphopeptides. at the end of this, what you get out is a really nice MS spectra, which can show the relative intensity of peptides between these three populations. And these are separated out because of the presence of those different labelled amino acids. So doing these experiments in Vancouver, I was able to do 3 biological replicates in my time there. And from those 3 experiments, we were able to characterise a total of 9,000 phosphopeptides, of which 7,000 were toxophosphopeptides. You can remember that these are in parasites which are growing inside host cells, so you're inevitably going to have some host cell contamination when you're taking a parasite inside a host cell, stimulating egress and hoping to clean out those toxopeptides. So, first of all, to look at the peptides which when you stimulate egress, are phosphorylated in a wild-type context, but which are not phosphorylated in a knockout context. So here what you'd hope to see is, for some peptides, you'll have essentially a 1-to-1 ratio of your phosphopeptides between the wild-type and heavy population and knockout heavy populations. But for some phosphopeptides, you'll see that it's dramatically upregulated in wild-type parasites. And so doing the analysis of this data, we're able to get almost 500 phosphosites characterised as being downregulated in this way. And this included a few interesting candidates, like another CDPK, CDPK2A, which we know is important for parasites, but we still haven't been able to show how. A sodium hydrogen exchanger, NHE1, which is known to be important for host cell egress based on previously published data from another group. We're also able to see that knockout parasites weren't upregulating phosphorylation of a guanylate cyclase or PKG, both of which are obviously involved in PKG signaling. Guanylate cyclase is the enzyme which produces cyclic GMP. And you also saw a couple of interesting proteins like a rhomboid protease here, which is known to be important for gliding motility of parasites. What we're really interested in is not those phosphosites which are just downregulated in knockout parasites, but what we really want to see are those phosphosites which go down in knockout parasites, but which then go back up, which then get re-upregulated when you also knock out TM5. So when we do this, when we have knocked out CDPK3, knocked out TM5, and restored egress in these parasites, we suddenly have a much smaller sample set to play with, which is fortunate because that's far too many phosphosites to try and play with. And as it is, this is too many. So 144 phosphosites corresponding to about 114 proteins. And these corresponded to 4 kind of main groups, including proteins with transporter function, vesicle transport, cell signaling, and also proteins which are already known to be involved in egress and host cell invasion. So looking at those which have transporter function, we're able to characterize phosphosites from a couple of major facilitator family transporters. We were able to characterize phosphopeptides, one phosphopeptide from a facultative glucose transporter, which is an interesting result. Obviously, you need glucose if you're going to be gliding around trying to find host cells to invade. And also, we again saw an interesting candidate in that sodium hydrogen exchanger, which is known to be important for egress already. So this is a really nice candidate right here of a phosphosite, which is probably really important for host cell egress. As for proteins involved in vesicle transport, it saw 2 Rab-like proteins and a SNAP protein. Sorry, a snare domain containing protein and a SNAP protein, all of which are involved in fusion of vesicles and the recycling of the enzymes involved in vesicle fusion. This is something which obviously be essential if you're secreting these microneum organelles, if you're secreting from these vesicle organelles all the time. So these are really interesting candidates right here also. In terms of proteins involved in cell signaling, there are a couple of interesting kinases, including a tyrosine kinase-like protein. Interesting because most apicomplexans aren't supposed to have tyrosine kinases. So that'll be interesting to look at further down the line, as well as a couple of peptides from a casein kinase. a phosphatidyl, a phosphoinositide phospholipase PIPLC, which is known to be involved in calcium signaling in other systems, as well as another kinase here with a plexigen homology domain, which is required for membrane localization. Again, membrane localization seems to be really important for these kinases. And we also saw 3 different phosphodiesterase, those enzymes which break down cyclic nucleotides like cyclic GMP, which are known to be important for things like PKG signaling. So these are going to be cool candidates going forwards also. And finally, these 3 proteins here, AMA1, MYC8, and RON9, these are all components of either the microneme organelles or the rotary organelles, which are probably not direct substrates of CDPK3. These are probably being phosphorylated after release from these organelles or during release from these organelles. But seeing these in our analysis is showing quite nicely that our criteria are working, that we are pulling out what we'd expect to see, proteins which are not being phosphorylated properly because parasites aren't egressing properly. But what we did see is a myosin which is thought to be involved in parasite motility and host cell invasion. So going forwards, we've got all these lovely little candidates to go ahead with. And so work is now progressing, trying to make knockouts of these proteins first off, and then looking at the importance of their phosphosites that we've characterised there in complementation studies. So to try and summarise, I've told you that Toxoplasma is a useful model organism for understanding other apicomplex and parasites. And really what's separating these parasites, as I mentioned, is the host cells that they recognise and the receptors that they recognise. But most data to date suggests that the signalling cascades regulating these motile stages here are essentially conserved between the parasites. So I've been able to show you that in recent years, we've been able to go from a model where we essentially didn't know anything about what happened between calcium release and motility, to knowing that CDPK1 was important in microneme secretion. We've been able to characterise that CDPK3 has some kind of amplification role in microneme secretion and motor activation, and that one of its targets is probably a protein called TM5, which we're able to pull out with that random chemical mutagenesis. Into all this, protein kinase G signalling has to work, probably acting to release that original calcium signal, into which CDPK4 probably has a synergistic role alongside CDPK3. And mixed into all this are a whole set of new potential candidates for being involved in this pathway. So the story is getting more and more exciting with each experiment, it seems. So that's more or less it for me. I'd obviously like to thank my supervisor, Chris, for trusting me with this really big project for the last 4 years. Alex Ubaldi is working very closely with me for a lot of this stuff. Fantastic postdoc in the lab, including all other members of the Tonkin lab are obviously fantastic to work with, just really great guys. And other members of infection immunity, while I'm at it, they know who they are. I hope they know what floor they're on. And every one of them is important in some way in keeping this a decent place to work. My advisory committee, including Juzzy, is my committee head. The sequencing bioinformatics was done by Tony and Jan. Some initial proteomics work has been done by Tom Nebel and Andrew Webb. Imaging facility, obviously, Gil Van Duren from the Australian National University helped make the original knockouts. And the sequencing of our mutated parasites was done over the AGRF by Lauren. And at the University of British Columbia, they hosted me for 3 months, or almost 3 months, and they were fantastic, with the lab head being Professor Leonard Foster. And thank you.
Thanks, James. Any questions on this stage?
So, two questions. Did you find TM5 as one of the differentially phosphorylated proteins in your screen? And secondly, was there, we're not surprised there's not a phenotype of the TM5 knockdown?
Yeah. Yes and yes. So there are some phosphosites which we see which overlap really nicely with our collaborators' suggested phosphosites using that substrate capture approach. It's a little bit inconsistent. In some of the replicates, they show really nice differences between the wild type and knockout parasites. In some of them, they don't. So that's something we're still picking apart now as to what that actually means. In terms of, are we surprised we don't see a phenotype? Not necessarily. The more we go on with these studies, the more we learn that, one of the great things about working with Toxo in the lab is it's extremely hardy. Like I mentioned, you can do almost anything with it. You can see how many different mutants you can make with it. You can leave them in the bin for a week and they'll come back up if you realise you don't have any frozen down stocks. But what that also means is that it's kind of hard to pick apart more subtle assays without the right tools. So if you're trying to, there seems to be either a lot of redundancy in these pathways or the parasites are just very, very tough. And our in vitro systems aren't particularly good at keeping them in check. And what you really need is to do as much stuff as you can in vivo, which obviously isn't feasible when you're doing this kind of large scale stuff. Oh, yes, sorry, good one. What is interesting is that in some cases, the removal of TM5 does actually seem to make parasites more sensitive to certain stimuli. So I mentioned that we hypothesised that TM5 was acting as some kind of suppressor on egress signalling. So what seems to happen is when you remove TM5 and then you stimulate with that Zapranas drug, which I mentioned before, which stimulates PKG signalling, these parasites are actually more sensitive to the Zapranas signalling, including CDPK3 knockout, TM5 knockout parasites, as in the double knockouts. So that's an interesting thing there, is actually they're actually more healthy, if anything.
So in your phosphoproteomics experiment, you said that you wanted to see, you were particularly interested in the phosphorylation sites that came back up in the double knockout. And then we were talking about direct targets and indirect targets. If they come up in the double knockouts, then CDPK3 can't be absorbed with the kinase. It would have to be indirect.
Yes.
But among the ones that were just different between wild type and single knockout, you had actually PKG itself.
Yeah, that's right.
Isn't that, if that affects its function, wouldn't that be enough in a sort of a very linear path towards CDPK1?
Yes. And so that's an obvious phosphosite to follow up on. It's interesting to think, the question of what's doing the phosphorylation when you don't have CDPK3 there and you don't have TM5 there, what's doing the phosphorylation is an interesting question. And that's something we're going to have to follow up on with all this stuff. But the PKG and those ones which are just knocked down in the knockout parasites, and the CDPK3 knockout parasites will still be interesting targets. The downstream stuff, the more kind of, in some ways, the more broad signalling proteins, what we really need to do is flesh out these pathways right now. So they're what we'd really like to know about. Well, both. We can't choose.
James, I'm getting back to TM5 and trying to ascertain its function, which I'm sure your lab's doing too. Have you tried to overexpress it, either on the membrane or solubilely? This is part A of the question. And part B was with EMS, you identified a mutation at C terminus, or near the C terminus. Is that a point mutation?
Yeah, they're all, so EMS just introduced point mutations.
Yeah, but it wasn't a stop?
No.
So have you made that mutation alone and validated it, and then expressed that as a negative control for your overexpressing?
So the first one about overexpression. Yes, we have tried the overexpression. Alex Ubaldi has done that work in the lab, and that hasn't shown any significant phenotype. The parasites seem to get along just fine. In terms of that point mutation in the C terminus, depending upon what kind of alignment you use, that seems to sit within one of the C-terminal transmembrane domains. So it's probably disrupting its targeting to the plasma membrane. And that is something we do need to continue on and do the complementation studies. That's something that's currently in progress, and that's something, we'll definitely be doing.
what we might expect is if you can't phosphorylate TM5, if you mutate those sites, then can't relieve its suppression. And that might be the way of getting at it, rather than just overexpressing.
Matt?
So in addition to the idea that PDK3 yeah, I'm not going to say it anymore, might be a direct regulator of egress through a substrate like TM5.
Yeah.
You also you proposed earlier on that it's sort of a more general calcium amplifier, which makes a lot of sense given your kinetic data. So how is that view fleshing out now from your proteomics approach as a general calcium amplifier?
Yeah, it's kind of it's stayed consistent. The tricky thing with phosphoproteomics is you're always going to have non-specific phosphorylation. So when you're seeing a nice number of even in that really nice specific substrate capture approach I showed, when you're that's unfortunately done in that was done in vivo, but in the past it had been done in vitro. So when we saw phosphorylation there, is that some non-specific off-target effect when you're taking the kinases outside of their natural context? So all that is to say that we do seem to see a lot of phosphorylation events happening, which are different between wild type and knockout parasites. So it does seem to support a more kind of general role for CDPK3 and egress. One of the things which I didn't show here, which was kind of interesting, is that we actually see a lot of proteins which you would think to be involved in transcription and things like that, which is probably partly to be expected because these are fairly abundant proteins as it is, so you're just going to pull them down with proteomics, but they're very consistent differences in their phosphorylation. So does that mean the parasites are actually kind of slightly accommodating this loss of CDPK3 in some way? And so with CDPK3 having such a broad effect that the parasites are actually kind of trying to work around it? It's going to be an interesting question going forward, but beyond the scope of what we're talking about now. So simple question, yes, it's bearing out.
Okay, we better finish there. This is only one of numerous stories that James has to finish off by the end of the year. So let's rally together and congratulate him on a great talk and finishing his PhD.